Hi, everyone. How are y'all doing? Good? Good? Enjoying your PyCon so far? Everybody going to go back to the hotel and nap or something? Come on. Anyway, we're about to have a fascinating talk. Trevor Bicoli. Oh, I killed it.
That's all right. Bicoli?
Thank you.
Yeah.
I promise I practiced that for like 5 minutes and I still messed it up. Anyway, he is a theoretical neuroscientist. That is a real thing and it sounds awesome. And he's going to talk to us about how to build a brain with Python.
All right. Thanks. So today, I want to tell you about your own brain. So the human brain, it's really an incredible computing device. It weighs about 2 kilograms. It's composed of 86 billion neurons and about a quadrillion connections between those neurons. It consumes only about 20 watts of energy, and yet it's responsible for everything that humans have created, art, language, computing, Python, which is why we're all here today. So I'm a theoretical neuroscientist, and what I do is I build models of the brain, I build computer simulations of the brain, and today I want to tell you how we use Python to make those simulations. So it's worth first starting off with why we want to simulate the brain. The first reason is that we want to understand it. So the late Richard Feynman, shortly before his passing, wrote this on the blackboard: "What I cannot create, I do not understand." So our opinion is that if we want to understand the brain, the way that we're going to attack it is to try to build a model of one to replicate the function of the brain under the same types of constraints that the biological system is under. The second reason that we want to create a model of the brain is to build better artificial intelligences. So there are pessimistic and optimistic examples of AIs in science fiction, and hopefully today I can convince you that these pessimistic eventualities are not that likely, partly because it's very difficult to engineer these types of intelligences. It takes a lot of work, and so this type of emergent superintelligence is very unlikely to happen in practice. So the types of software that I'm going to tell you about today are called neural simulators. There's 3 very important features of neural simulators that I'm going to talk about. The first is that they attempt to emulate neurobiology at some level of abstraction. And as a consequence of emulating neurobiology, they operate in continuous time and they communicate with spikes. So I'll talk a bit about what that means shortly. But I bring up these 3 kind of rules as a way to differentiate neural simulators from other types of artificial neural network programs. So you may have heard about these kind of massively huge deep learning systems that Facebook and Google are using to process images and speech. That's not what I'm going to be talking about today. These systems don't attempt to emulate neurobiology. In particular, they work in discrete time rather than continuous time, and they don't communicate with spikes. So the first neural simulator that I'm going to talk about is appropriately one of the oldest, yet it's still being actively developed and in use today. It's called Neuron. At its core, it's a C program with a graphical interface with some Python bindings. And so let's start it up. There's a little thing that it comes with called the Neuron demo. So let's start up the Neuron demo. Like I said, old program, old GUI. So I'll load up this pyramidal example to give you a little bit of a sense of what I mean by neurobiology. So this is essentially a simulated neuron. All of these processes around here, except for one of them, are called dendrites. And you can think of dendrites as providing input to this cell. And they provide their input as current. All these dendrites converge upon this center point, the body of the cell, which we call the soma. And out of the soma, there will be one projection that provides output to other cells, which we call the axon. So essentially, it's an oversimplification, but one way to think about a neuron is essentially as a function that takes in inputs from its dendrites and provides a single output through its axon. So what Neuron is doing right now is called a current clamp experiment. So this I clamp here, I stands for current. And essentially at this blue dot, there's a simulated electrode that's injecting a little bit of current into the soma of this cell. So if I run this model now, over here, this graph populates. On the x-axis, we have time in milliseconds. On the y-axis, we have the voltage recorded from this cell. And just in this kind of screenshot, we have the three features of neural simulators that I was talking about. So we're emulating neurobiology. We're working in continuous time. There's no discontinuities in this graph. And this upwards and downwards stereotyped voltage trace is called a spike. So if we move this, this current clamp to another part of the dendritic tree, we run it again, we no longer get a spike. So it's still, some current is being injected, but it doesn't spike. Spikes are the single way that neurons communicate, at least in this oversimplified picture. When a cell spikes, that spike travels down the axon and communicates a little bit of information to the downstream neurons. So as you can see, there's lots of things that we could do in this simulation, lots of variables we could play around with. And so I'm going to play around with those a little bit in Python, using the Python bindings of Neuron. So this is going to be a simplified cell, just has two parts, a soma and a single dendrite. Essentially what we're doing here is setting some parameters. The length of our soma is going to be 40 micrometers and 20 micrometers in diameter. We're going to insert some ion channels into there. The dendrite is going to be 150 micrometers long and 3 micrometers in diameter. It has a different set of ion channels connected up together. And then to simulate our current clamp experiment, we make this iClamp object. We give it the thing that we're clamping the current of. We're going to inject, in this case, 1 nanoamp of current for 500 milliseconds. And then what Neuron calls vectors are how we're essentially collecting the data from that experiment. So we can initialize the voltage in this cell, and then run it for 500 milliseconds. We can take the time and the voltage and put them into NumPy arrays, and then use matplotlib to create a plot of that membrane voltage over time. So in this 500 millisecond window, it spikes quite a bit. There's a lot of information being transmitted from this cell to its downstream cells. So you might be wondering how this simulation actually works. What's kind of the math behind it? I won't go into that in very much detail, but I'll say that Neuron has built into it a number of mathematical equations that essentially emulate electrical physics. So we think of a neuron as kind of like an electrical device. And there are more precise and less precise ways to emulate those physics. If you wanted to write your own neuron model that is, either a more or less precise version of what Neuron has built in, you can write it in this language. It's kind of a domain-specific language for writing neuron models. Unfortunately, it's not Python. It's actually, not the worst language. If you're a theoretical neuroscientist, all these things make sense to you. So, so it's not too terrible a language, but it means that there's a two-step process for running your model. You have to compile this, then run your Python script, and that's not great. And so there's other neural simulators. The next one I'll talk about is called Brian. Brian is a neural simulator implemented entirely in Python, and it puts the equations behind these neurons kind of front and center. So an example Brian script to create a neuron model kind of has at its core this string which defines the equations of these cells. So a complicated equation, don't expect you to actually understand it, but it's just a string in that Python script which gets regenerated every time we run it. Up here, we're just defining a bunch of parameters, and this shows off another nice feature of Brian, which is that it explicitly tracks the units associated with these parameters. So this reset voltage, for example, is negative 70.6 millivolts, which is very important for validation of models, making sure that you're doing the right thing. So we can create what Brian calls a neuron group with those equations. We're just going to make one neuron in this case. We set its membrane voltage to that reset voltage, inject 1.5 nanoamps of current, and when so we can then we monitor the voltage from that cell and the spikes coming out of it, run it for 500 milliseconds, and we get something that looks very similar to what we had in Neuron. So across this 500 millisecond time window, we have a bunch of spikes. It kind of seems to slow spiking after a while. That's interesting, I suppose. But to me, it's not the most interesting thing. Because like I said, this talk is about how to build a brain, but so far all I've showed you is how to simulate neurons. And to me, those two things are kind of qualitatively different. And so the question is, how do we scale up these simulations from neural simulators, of which there are many? So these are 8 examples in Python, but there are actually quite a few more. How do we scale up these types of simulations to something that's brain-like and something that you could say has some kind of intelligence? So scaling from neurons to brains. People have been trying to tackle this problem for quite a while and have come up with a bunch of different options, which I'm going to poll the audience to see what you think is the most promising. So one option is to, so I should say, scaling up, we know that we need an 86 billion neuron network. You can just throw a bunch of computers at that. That'll happen eventually. That's fine. That's no problem. But it's the 1 quadrillion connections that's really the problem. Because how do you figure out how to connect these neurons up together? Each connection has a weight associated with it. How do we figure out what are good weights? So the first option is that you could just randomly generate a whole bunch of weights. And if you really believe that the neuron is essential to what's happening in intelligence, it might be the case that connecting them up randomly is sufficient. The second option is to do some recordings from brains to figure out the connectivity patterns, the statistics of connections in the actual real brain. And people do this in a field called connectomics, and you basically get an idea of what's connected and the relative strength between those areas. The third option is to connect things up functionally. So neuroscientists have essentially ascribed functional functions to a bunch of brain areas. So at the back you have visual areas, areas that process visual information, motor areas, kind of working memories out the front here. One way we could scale would be to kind of take a region of the brain and try to replicate its function in a brain-like way. So let's get a little quick show of hands, even though I've highly biased this crowd. But who thinks that option 1, connecting these neurons randomly, is might result in human scale intelligence? Hands up for that. Good, we got a few hands. Nice. Okay, what about option 2? Who thinks that the statistics are the most salient point and connecting them up that way will work? Also quite a few hands. What about option 3? Hands up for if you think that the function is the most important part of things here. Apparently I did not bias you hard enough. So it seems that there's kind of an even split between statistics and function. In my lab, our Crystal Lassimus lab, we're really dedicated to scaling things on a functional level. So we're interested in how are these neural networks, how are these networks of spiking neurons representing information, transforming that information, and how can we use that to build essentially functionally relevant brain net models. So the tool that we've created to do this is called Nengo. I should say that it's possible to come up with these functional networks in a very kind of precise methodological way of varying parameters and seeing if you get functional networks. But we think that tools are tools really needed in this space. So the tool that we created is called Nengo, and it connects neurons together using the principles of the neural engineering framework. And just like the other neural simulators I've talked about to this point, it attempts to emulate neurobiology. It works in continuous time, and it communicates using spikes. So to this point, I've been talking about neural spikes, and I've been showing you voltage traces. And I want to, we're talking about scaling up. And so the way that we scale up those voltage traces are to plot what we call spike rasters. So each of these vertical ticks represents a spike coming from a neuron. In our case, across the rows, we have individual neurons. So this row shows you the spikes that this neuron emitted over time. And there's clearly some pattern to this spike raster. There's clearly a lot of activity around this part among a certain subset of the cells, and a lot of activity over here for the other subset of the cells. And so how do we make any sense of this? How can we use these the neural firing patterns to represent information? Fortunately, neurons don't respond in an entirely arbitrary way. We can kind of characterize the way that a particular neuron responds with a tuning curve, which is what this is. So imagine that you're recording from a neuron that's downstream from the sensory part of your fingertip, your right fingertip. If you put a little bit of pressure, the nerve endings in your finger will send current down to that neuron, and the more pressure you put onto it, the more current gets injected, and the more this cell will spike. So if you record this a lot, you can kind of average this out, and you get a curve like this. So if you're recording from this cell and you know that it's spiking at around 200 hertz, you can kind of make the judgment call that it's representing a pressure amount that is kind of 0.1 on this scale. Of course, this is an idealized version of what really happens. Neurons are extremely noisy. You can record from the same neuron twice, and you'll get, slightly varying results. And so we really need to combine the responses of many neurons together to get an idea of what's happening. And so the way that we do that in Nengo is actually very analogous to binary coding. So, in computing, binary coding is how we represent information. If we wanted to represent, for example, the integer 13, we know that we can encode that as the binary string 1101. Each of these bits, each of these binary digits represents a different power of 2. So at the low end, this 1 represents 2 to the 0 or 1. At the high end, this 1 represents 2 to the 3rd or 8. And knowing, what's true and false and what power of 2 these bits represent, we can essentially do a weighted linear sum. So we weight the true or false value by the power of 2, add that all up, and we get back our originally encoded integer 13 as our kind of decoding. So we're essentially encoding this integer into binary and decoding it back out. In binary code, this happens perfectly. Your computer never gets this wrong. They're pretty much always right. The neural code, analogously, if we're trying to encode a number like 0.5, this might represent kind of medium pressure on your finger. The neurons that are sensitive to that quantity are going to spike at a certain rate. So say we have a neuron that's spiking at 23 hertz, 60 hertz, 3 hertz. Nengo will figure out a set of decoding weights, D, associated with these neurons such that we can do the same weighted sum to get back an estimate of that originally encoded value. So 23 times our first decoder, 60 times our second decoder, et cetera, et cetera. You add all these things up, and you get a number that's close to 0.5. It's not exact, but in most cases, it's sufficient. So what essentially this gives us is a way to represent information, and we can use similar methods to transform that information. And essentially what this allows us to do is to work on a higher level of abstraction than if we were just making neurons and connecting them together individually. So a good analogy for this is our compilers in computing. So you can think about this little snippet of C code, pretty understandable what it does, but if you go down to the assembler level, it's quite difficult to understand. And even this is just kind of a shorthand for binary instructions, also very hard to understand. And I think you can agree that if we were programming in assembler, we wouldn't be at the state of computing that we're at right now. We certainly wouldn't be at a Python conference, because it's another language. Nengo, analogously, is like a neural compiler. So we specify a neural model in terms of what information or what values are being represented and transformed. So up here we have a node. A node is how we represent non-neural information. So kind of like the current that we injected in that current clap experiment, we need a way to somehow start off our simulations. So we call them nodes. Ensembles are how we represent information with populations of neurons. And so when we connect them, in Nengo, when you connect from a node to an ensemble, we're essentially figuring out how much current needs to be injected into that population to represent the value that your node is encoding. Similarly, when you connect two populations together, in this case x and squared, we figure out the connection weights between them such that they transmit that information. And you can also transform that information across that connection. So in this case, we're going to square the value in x and transmit that to squared, hence the names. And under the hood, Nengo generates both randomly and through some optimization processes a bunch of numbers that would be a huge pain to figure out by hand. So the gain and bias on these neurons, which tells you how much current to inject, the decoding weights on this connection to actually implement that squared function. So let's look at this happening in Python code. Nengo is a Python library. So we take those nodes and ensembles from before, we group them together in an abstraction called a network, and then we add some probes in order to collect data over the course of this simulation. We create a simulator object, so nengo.simulator, pass it the network that you want to simulate. We run it for a second, and so let's look at the things that we've probed in that simulation. So the first thing is the voltage coming out of just one of the neurons in the X population. You can see that just like in the other simulators, we're tracking very similar types of information. We can look at the voltage trace of this cell over time. We can also look at the spike raster. So thinking about all of the spikes that came out of our X population, there's a pretty clear pattern here because we're just representing a constant value 0.5 that all the cells are kind of spiking at a regular rate. But since we have this higher level of abstraction in Nango, we can also look at the value that these groups of neurons are representing over time. So our X population, the blue curve, very quickly reaches a value of 0.5, which is what we were trying to encode in the first place. And our squared population, the green curve, encodes X squared, which is about 0.25. We're doing a pretty okay job here. I should say it's not doing the best job, because what we've asked this network to do represent just one value is kind of an odd thing to do with these types of models. Like I said, they operate in continuous time, which means that they're kind of dynamic systems. And so it makes they work a lot better in dynamic situations. So what I could do is instead of having the output of my value node be a single value, I can change it to be a function of time. So in this case, I'm just changing the output of my value node, my val node, to be a sine wave. I simulate that network again. If we look at the voltage trace in that neuron in X again, there are some areas here where it's not getting any injected current, and so the voltage trace is kind of staying at a minimum value. If we look at the spike raster, we see now that there's kind of a clear pattern happening here, that we have some cells that are sensitive to the start of the sine wave and other cells that are sensitive to the end of that sine wave. And if we decode out that value, we get a pretty good representation of the sine wave that we were trying to encode. So we get the up-down behavior in our X population, and we represent the square in this X squared population. So essentially, you can take these two kind of ideas that Nango is a neural compiler that uses a type of coding that's analogous to binary coding, a couple other ideas, and like 10 or so years of research, and you can build models that work very functionally like the brain. So this is a model that we published a couple years ago called Spawn, and in it, we kind of figured out functionally relevant networks for vision, for motor control, working memory, all these types of things. And we put all this together in such a way that it can perform 8 cognitive tasks. So this is it performing one of those tasks. And you'll see it write its answer at the end here, 555. That's the right answer, in case you were curious. I don't have time to go into the detail about Spawn, but if you're interested, I encourage you to check out nango.ca for some videos of Spawn happening. You can read our paper. But what I want to highlight here by showing Spawn is that Nango allows us to scale things up in an exciting way. One of the ways in particular that we enable this type of scaling is with this new version of Nango that we just released. It took a lot of weeks of research and kind of tweaking to get Spawn to work. One of the components that was quite difficult is called the basal ganglia, and it essentially does action selection in Spawn. But now, with this new version of Nango, anyone can pip install Nango and get the same basal ganglia in their models that we have in Spawn in just one line of code. Similarly, we've been working on ways of scaling up that are specific to different types of computing platforms. So you can get this Nango OCL package, which includes a simulator that will run the exact same Nango model, but it will run it using an OpenCL backend, which means that you can run it on GPUs. We also have a backend that runs on what's called a Spinnaker neuromorphic hardware, which is an exciting project coming out of Manchester, University of Manchester. So these are all the ways that Nango is helping scale up to the brain, to kind of the whole brain level. And if you're interested in these types of things, you can go look at our GitHub organization, github.com/Nango. If you want to know lots of details, then I encourage you to check out my supervisor, Chris Elias Smith's book, How to Build a Brain. But I want to finish off showing you one of the things that we're working on lately, which is a way to kind of interactively visualize what's happening in these types of networks. So what's happening in this model isn't super important, but just know that there are two ensembles here. One with 1,000 neurons, one with 100 neurons. This one's connected to itself, that one's connected to that ensemble. And I'll start up the visualizer that we've been working on. So in here, we get kind of a miniature version of our network. So our stim node the oscillator population, the shape population. If I press play, it starts simulating. You can see that this is indeed working in continuous time. Time is just flowing across that x-axis. We're generating spikes, and we're representing values. I can give this a little kick in our stim, and now it represents a function that we've encoded here, kind of an exotic function in a two-dimensional space. So, what I hope to kind of prove by this simulation and other types of things is that, our original goal was to understand how the brain works and to build AIs. I hope this shows that, we do actually know quite a bit about how the brain works, and using those ideas, we've been able to create a Python library that allows you to use these biological parts to create pretty exotic functions, and you can scale that up to things like Spawn. But what we're working on right now is really the, more of the scaling part to build artificial intelligences that will help humans in the real world. So hopefully, in 5 or 10 years, I can come back to PyCon and tell you about those projects. But for now, I'll end there. Thanks.
All right, we have about 5 minutes for questions. If you want to go ahead and line up the microphone, if you have any question you'd like to ask, go right ahead.
Yes, I have a question. I know that there exists a tiny worm, the C. elegans,
Yes?
which has only 900 neurons, and
Yeah.
they connect. And how they are connected is already known. Have you tried to simulate that nervous system?
Yeah, so about C. elegans, not only do we know how many neurons there are, we know kind of how they're connected. We have a very detailed model, or sorry, I should say we have a very detailed idea, kind of neuroscientifically, about what C. elegans' brain looks like. There's a project called, so I should say that we haven't tried to simulate that ourselves, mostly because the types of neurons that we simulate are all kind of similar. So pyramidal cells in your cortex are all kind of similar, and so you can kind of treat them in a similar way. But in kind of invertebrates and other lower organisms, all of their neurons are very complicated. So you would really need a tool like Neuron that allows you to have a specific function for each neuron to make a kind of a C. elegans simulation. But there's a project called OpenWorm that's doing that, which is very cool.
Hi, thank you for your talk. I was wondering if I could ask a question about one of the basic assumptions of your model. You said that use the functional model. How does that take into account people who've lost part of their brain and have had other parts kind of compensate for that? Does that imply that brain location is kind of agnostic to the function?
Oh, yeah. So I should say that, so remember in those models, we were making kind of discrete ensembles of neurons, right? So, when we build an Ego model like this, it contains, maybe, 100 or 200 ensembles that are connected together, right? Those ensembles you would kind of hypothesize to be located in similar parts of the brain. So in Spawn, we have 1,000 or so, maybe thousands of ensembles. You can kind of say that each one of these ensembles is kind of part of a functionally relevant subnetwork, and that subnetwork you can kind of hypothesize to be in a particular region of the brain. So the parts of Spawn that are dedicated to visual processing, we would think of those to be in like V1 and V2. And you can do similar things. It's not as detailed as a neuron where you actually say, this is the spatial properties of the cell, but you can still kind of make analogies to, make hypotheses about where this ensemble might be in the brain.
Awesome.
Thank you.
Can we get the back of the room for a question?
You mentioned sci-fi.
Oh, hold on, sorry. Go ahead.
You mentioned sci-fi. So in your professional opinion, what's the best, most interesting AI in science fiction?
Yeah, that's a good question. I actually watched Terminator specifically for this talk because, it's happened a few times that people have come up to me and been like, so you're building Skynet? And I'm like, I don't think so, but I should watch the movie to find out. The problem with all these AIs in science fiction is that they're kind of underexplained. I just want to know more about the details.
Hence my question.
Yeah, great.
Any for the room?
Does this have general application to like machine learning, or as someone in the audience not trying to emulate a brain, is there some way I could use this in a more general application?
Yeah, so we're really hoping that that will be the case. But like I said, we've really been focusing on emulating neurobiology for now. We think that there are very important ways that this can be applied in industry. The really kind of killer feature of spiking neural networks is that they're incredibly efficient. So I mentioned that Spinnaker neuromorphic hardware, it can run on 3 orders of magnitude less power than typical kind of conventional computing. The problem is that they don't know how to program their chips because they're just neural networks. They're simulating neurobiological systems. So we're hoping that Nango is going to be a way to program those types of chips to do interesting kind of machine learning type functions.
Thanks.
Let's do the question from the back.
Thanks. Great talk. Thank you. Very interesting.
Thanks.
One question I had is, it seems like, for the most part, for the small systems, you sort of get back what you put in, which validates the code.
Mm-hmm.
But in terms of understanding or producing novel things, how complicated, how big do the systems need to get, and do you have to modify the code in other ways before you start to get sort of really meaningful functional behavior that you can ascribe to something similar to what a brain would do?
Mm-hmm. So I can tell you that you don't need to modify the code at all. So that's been really a big push for this new version of Nango, is that in the old version, we had to make a lot of modifications to get Spawn running, but now you can basically take Nango 2 as it is, and you can run Spawn. As far as how to build these models, it is really an art. It takes a long time to kind of think about how you take basically time-varying vectors and functions on those vectors, and with that kind of compose it all into a brain-like system. And if you're interested, you should definitely come work in my lab. But if you go to the GitHub page for Nango, in the documentation, we have a long set of examples that kind of go from a simple network to a very complicated network. And if you look through those examples, you should hopefully get a sense of kind of what that art is like.
Excellent.
Thank you.
Let's take 2 more questions from the front of the room.
All right, thanks for your talk. Your system, you did a good job of demonstrating modeling at the neural level and the functional level.
Mm-hmm.
Does it have any provisions for modeling at some of the intermediate, like the cortical column level or some of the other circuits?
Right. So, so I would say that we're right now kind of at that intermediate stage, because we've kind of focused on, what are the functionally relevant subsystems, and then make a very reduced model of that, and then connect that together. So Spawn only has 2.5 million simulated neurons, which is, less than 0.1% of the real brain. So we've just kind of chosen little modules, and so if you wanted a more sophisticated visual system, you could kind of take the visual system that's in there and just scale it up kind of in a obvious fashion of just increase the dimensionality, be more sensitive to more pixels, all that kind of stuff. So what we have now, and this abstraction that we call a network, is essentially that middle ground where it's not an ensemble, it's like a group of ensembles, and that group of ensembles together compute kind of an interesting function, more interesting than just something that is in one layer.
Thank you.
Hi, I've been told that the power of the brain was the capacity of reconnection of neurons. And so I'm just wondering, have I been lied to, or? And the second question is, why don't you spawn a random brain and just ask for reconnections?
Sorry, can you expand a little bit about what you mean by the power of the brain is?
So I thought that neurons had the power of disconnecting and reconnecting between each other.
Oh yeah, so one thing that I haven't talked about at all in this presentation is learning. So learning is really, how our brain works, how it goes from, development all the way to a fully functioning adult. So we do have, the capacity to learn in Nango. What I presented to you today is all based on kind of optimization methods, and what it gives you is essentially kind of a picture of the end result of learning. So there's a lot of work to do in figuring out ways to get from kind of just a blob of neurons to figure out error signals such that you can kind of make these really complicated networks. That's kind of a separate line of research.
So I guess my question is more, why can't you just pound a random brain and make it learn from scratch?
Definitely people are trying to do that.
Okay.
But I can tell you that they haven't come up with anything as kind of functionally significant as what we've done with Nango.
All right.
Okay,
Thank you very much, everyone, and thank you, Trevor.
Thank you.
Just a reminder, this is the last talks of the day. If you're going to one of the awesome PyCon dinners, you're probably going to have to book it on over there. Have a great evening, everyone.